Namaste students, I am JV Dr. Ashish Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Vertical Science, Jayoti Vidyapeeth Women's University, Jaipur. Today I am going to discuss about a topic, bacterial classification, and this is the continuous part of the previous part of bacterial classification, in which I discussed about the classification of the bacteria on the basis of their morphology. Today I am going to discuss about the bacterial classification on various different bases. And the second basis of bacterial classification is the capsule, presence of the capsules. So on the surface of the bacteria, there are some surface structures. There are some surface structures, and the availability of these surface structures bacteria can also be classified. Some of the bacteria have a specific outer covering outside the cell wall, which is known as the capsules. These capsules have an important role in providing support to the bacteria so that they can attach to any kind of surface. These capsules are responsible for the pathogenic nature of the bacteria. So, if capsules, some bacteria have capsules and some bacteria do not have capsules. So, if the capsules are present on the bacteria are known as the capsule. Bacteria which do not have the capsules are known as the non-capsulated bacteria. The example of the capsulated bacteria is the Streptococcus pneumoniae. And the examples of the non-capsulated bacteria is the Viridans streptococci. So this is the one type of classification. The second one is presence of the flagella. Flagella are also there, which are present on the surface of different microorganisms. In the bacteria, they are also present. Bacteria, these flagella are also the surface structures. And if the flagella are present in the bacteria, then they are known as the flagellate. And if the flagella is not found on the surface of the bacteria, they are known as the aflagellate. Flagellate means the flagella is present on the surface of the bacteria, so the bacteria are known as the flagellate. And aflagellate means the flagella is not present on the surface of the bacteria, so they are known as the aflagellate. So if we talk about the flagellate, the presence of the flagella on different areas can also be used for the classification. Uh, the first one is a monotrichous. If the flagella is present on one end of the bacteria, then it is known as the monotrichous. Monotrichous, if the flagella is present in different numbers in one end, means the bacteria is present on the flagella is present on the one end of the bacteria, but they are not single, they are multiples. So they are known the um, lopotrichous. If the flagella is present on both ends of the bacteria, then they are known the amphitrichous on both ends. One single on both ends, they are known the amphitrichous. And if the flagella is present on the all the surface of the bacteria, then it is known as the Vegetarians. So flagellate are also divided into four more groups: Slopotrichus, Amphitrichus, and Peritrichus. Presence of spores is also a basis of the classification of the bacteria. Some bacteria have the capability to produce spores. What is the role of the spores? Spores are the some specific part of the microorganism make them to survive in the harsh conditions, which make the bacteria to survive in the extreme environment. For example, the extreme environment like high temperature, starvation condition when the bacteria do not have any nutrients and they are starving. So, in these conditions, some bacteria can produce spores. So, on the basis of the production of spores, bacteria are divided into two groups. spore forming bacteria and non-spore forming bacteria. Spore forming bacteria is example. The example include the Bacillus species. The non-sporing bacteria have the bacteria which do not produce any kind of spores, and examples is the E. coli. Now, classification of the bacteria on the basis of uh, staining. On the basis of staining, if we talk about the identification of the bacteria, for identification of the bacteria, we need to observe them on the microscope. But if we directly observe them in the microscope, we cannot find anything because the bacteria are not visible to eyes due to the color variability and the transparency of the color. Because they are naturally stage, and if they are not providing any kind of color, then they cannot be identified. So some special stains, some staining dyes are used so that they can be used to stain the bacteria and we can identify the bacteria in a particular kind of color. So bacteria have some specific organelles which can absorb these dyes, these staining dyes. And, and when they absorb these staining dyes, they can be identified. They are identified on the basis of their color, violet color, pink color, but whatever their color can produce. So they are identified on the basis of their particular color. So on the basis of their staining, we can classify them. So what are their classification patterns? So first one is the gram staining. Uh, Christian Gram developed a technology for the identification of the bacteria. The gram positive and gram negative are the two types of bacteria which are present. So Christian Gram, on the basis of the gram staining, we can identify the kind of bacteria, two classes of the bacteria. First one is the gram positive and the second one is the gram negative. The gram positive are the bacteria which are identified when the color of the bacteria are in violet color. So if we observe in the microscope, the bacteria which is found in the violet in color after giving the gram stain, then they are known the gram positive bacteria. And the gram negative bacteria are the bacteria which are observed in the pink color, okay, which are observed in the pink color after giving a saffronin dye, then these type of microorganisms are known as the gram-negative bacteria. The differentiation between the gram-positive and the gram-negative bacteria is dependent on the thickness of their peptidoglycan layer. Their peptidoglycan layer in the gram-positive bacteria is thick, and peptidoglycan layer in the gram-negative bacteria is thin. So, the gram-positive bacteria absorb the dye quickly, and gram-negative do not absorb the crystal violet color. So, they do not appear in the violet color. Next type of staining is acid fast staining. Some organisms have the ability to identify to be identified by on the basis of the gene Nelson staining. If the gene Nelson stain is occupied by the bacteria, then they are known as the acid fast staining. Acid fast organism, and if they do not capture the acid fast stain, then the bacteria are known as the non-acid fast organism. So acid fast organism is pink in color, and non-acid fast forming bacteria will appear in the blue color, so they can be identified. Classification of bacteria, talk about the examples of the gram positive and gram negative and the acid fast forming bacteria. So the example of the gram positive cocci is the Staphylococcus aureus. Example of gram negative cocci is the Neisseria gonorrhea. The example of the gram-positive roots is Clostridium. The example of the gram-negative roots is the E. coli. The example of the acid-fast bacilli is the Mycobacterium tuberculosis. The example of non-acid-fast bacilli is the Staphylococcus. So these are the basis of the classification of the bacteria on the basis of their uh, on the basis of their staining. Next is the classification of the bacteria on the basis of their pathogenicity. We can classify the bacteria bacteria on the pathogenicity because some bacteria have the capacity to cause disease. Bacteria which can cause disease into the humans, they are known as the pathogenic in nature. So, on the basis of the pathogenicity, and the bacteria can be divided into three groups: pathogens, non-pathogens, and commensals. Pathogens are the organisms which are able to cause and spread the disease in the humans and some microorganisms in humans and some plants and some animals. So these are known as the pathogenic bacteria. Non-pathogenic bacteria are the bacteria which do not cause disease. For example, is the lactobacilli and commensals. And normally they are non-pathogenic in nature, but sometimes they show the disease when immunity is impaired. They are not usually pathogenic, but sometimes can be pathogenic when the immunity is impaired. So these are the classes of the different kinds of microorganisms. So this is the in this lecture I discussed about the classification of the bacteria on various bases, uh, like classification of bacteria on the presence of different kinds of characters, like presence of the capsule forming bacteria and non-capsulated bacteria, flagella, flagellate and non-flagellate flagellate bacteria on the basis of the presence of the flagella, spore forming bacteria and non-spore forming bacteria on the basis of the presence of the spores. And on the basis of the staining, we also classify the bacteria on the basis of the presence of different dyes such as the gram-positive bacteria and gram-negative bacteria, acid-fast bacteria and non-acid-fast bacteria. Some another type of bacteria can be also classified on the basis of the pathogens like pathogenic bacteria and non-pathogenic bacteria and common cells bacteria. These all are the classes which I discussed in this lecture. So I will compile my lecture here. And the question which can arise here can classify the bacteria on the basis of their pathogenicity, their staining, and their presence of the some specific outer layers. So by these characters, we can easily identify and we can easily classify the bacteria. So my lecture is over here. If you have any query, you can ask me. So thank you very much.